Without further ado, our next speaker hails from Australia, and we're excited that she and her colleague came all the way over here to present. Dr. Joanne Fielding is going to give us a talk on visual snow, a behavioral perspective. Dr. Fielding.
Okay, so I'd firstly like to start by thanking Paul and Sierra for the invitation to speak here today. I feel incredibly privileged to be part of this inaugural event, and I'm certainly looking forward to collectively changing the visual snow landscape. Now, together with Professor Owen White, who you'll meet a little bit later in the program, I'm the director of an Ocular Motor Research Laboratory based at the Alfred Hospital in Melbourne, in Australia. And for the last almost 20 years or so, we've collectively been researching brain function. Firstly, with a view to understanding how the brain works, but more often than not, to determine what happens when the brain doesn't function as it should. And we've been primarily doing this through investigating the visual and ocular motor systems, so the systems that allow us to see and the system that allows us to move the eye so that we can essentially see more clearly. I've just popped in a gratuitous shot of Melbourne here. Apparently it's the world's most livable city and has been so for the last 3 years running. I guess like any major city, it depends whereabouts in the city you're living. But from my perspective, it's very livable. Okay, so I just want to start briefly by acknowledging the members of our research team that are actually involved in researching visual snow. We have a very large group, but these are the guys that I want to acknowledge. We're an interdisciplinary team with various research interests, comprising neuro-ophthalmologists. We have an ophthalmologist, but we also have a team of neuropsychologists. And our collective interest in visual snow is a fairly recent one. And I'm going to leave it to Professor White to let you know how this interest actually came about in the first place. And what I'm going to do is quite different to Jim, and I'm going to lunge in head first and talk about the research that we've been conducting, and particularly explain why we're using a behavioural perspective, which is not intuitive, and then have a brief look at the research that we plan to do in the future. Okay, I'm a research scientist, I'm not a clinician, and my research training is actually in psychology. Now, I didn't put that on the website because I'm acutely aware that a large number of you in the audience actually suffer from visual snow, and I'm also aware that the term psychology means something very different to different people. And I desperately wanted to avoid raising the spectre of visual snow appearing as a psychological condition. It's incredibly clear that visual snow is not all in the mind, it's very much a neurological condition. So for those of you who don't know what neuropsychology is, a quick definition. So we aim to understand how cognition and in turn behaviour is influenced by brain functioning. So we're not looking at aberrant thought processes, we're looking at aberrant brain processes, and that's quite different. And by cognition, I'm talking about that we process all the different forms of information that are available to us, not only from the senses, but from our experiences, beliefs and expectations, assimilating all of those processes and linking it to adaptive behaviour. And I guess at first glance, visual snow has nothing whatsoever to do with cognition. And certainly visual snow is not a behaviour, it's a perception. You'll have to bear with me so I can connect the dots for you. We might be starting to get an inkling or some sort of understanding of what might be driving visual snow in the first place. And collectively, the proposal is that of heightened neuronal sensitivity or cortical hyperexcitability, which you may have seen certainly in the literature. And certainly within the visual system, although there is various opinion about whereabouts in the visual system that may be, but obviously not necessarily just in the visual system if you think about the associated symptoms, including tinnitus, paresthesias, tremors, etc. But certainly understanding what these changes are is an necessary and important first step in treating visual snow. So somewhere in the visual system, up to 50% of the brain, and probably more than 50% of the brain, is actually involved in processing vision. So most obviously, the pathway that I've indicated in blue here, so from the retina all the way back to the visual cortices. But also there's a large part of the brain that is involved in vision in another sense, including the parietal regions of the brain towards the rear of the brain that allow us to construct spatial coordinates of the world around us, and very importantly, the prefrontal cortices, and they serve a twofold, they have a twofold purpose rather. The first of these purposes is to make sense of the visual information that's processed at the back of the brain, and the second is actually a top-down influence that shapes or changes neural activity in the back of the brain before we even perceive a visual image. So we've obviously got multiple regions that influence what we effectively see, and this is a very extensive and integrated network. Okay, I've constructed this diagram to give you some idea of the complexities or the interconnectedness of this particular network. So you can imagine it's not particularly easy to establish what's driving changes across the system in visual snow, or in fact to directly measure what are possibly very subtle changes in network function, and that's been the difficulty all along. The network actually looks something more like this, but will result back to a more simple diagram. Bear with me. The point of all this complex processing, however, is actually to guide adaptive or meaningful behaviour. It's not just to stand back and admire the view as it was, but to navigate and interact with our environment. And so these network changes can then therefore be measured behaviourally, provided that you ask the brain the right sort of questions. So we start with this idea of cortical hyperexcitability, but we also recognise the fact that this excitability might be a consequence of some sort of reduction in inhibitory control. So if you think about it as neurons firing wildly as opposed to difficulty dampening down the activity of neurons. And pitched this way, the cognitive domain of attention becomes particularly relevant. Attention effectively underlies all other cognitive processes. And by attention, what I'm talking about is the selection process that allows the brain to focus on what's important at any given time, so what is behaviourally relevant. So what it does is focus the processing of relevant information, as well as inhibiting or filtering out what is irrelevant, as I said, in any given context. This is a very crude diagram of the circuitry involved, but they are those prefrontal regions I was talking about, as well as those parietal regions. And they effectively create two interconnecting subnetworks that deploy attention firstly by virtue of a stimulus property, so what we actually physically see in the environment. There's a stimulus-driven or bottom-up allocation of attention, or according to our expectations or goals, that is a cognitively driven process or a top-down process. So just so that you know what I mean when I refer to top-down and bottom-up. Okay, so these prefrontal and parietal regions actually integrate the bottom-up and top-down information that I was talking about and collectively construct what I've termed a priority map, where a stimulus or a particular spatial location is represented neurally by a level of activity that reflects its attentional priority. And this signal actually modulates sensory processing in all the visual areas that I showed you before, so that things that are of interest are optimally analysed and distracting things are effectively filtered out. And this modulation is seen in all areas of the brain where you have visually responsive neurons. What I wanted to give a quick nod to was Jim's previous talk. And he was talking about the source of visual snow, and there's a significant overlap if you think about what he was saying earlier in terms of the saliency maps in the parietal lobe, the high-level cognitive circuits in the prefrontal lobe, as well as the thalamic reticular nucleus, which plays an important role in regulating the flow of information to and from these cortical areas. What's important to us in our lab is the fact that the networks involved in attention are actually the same as those networks involved in programming a Sicard. Now, a Sicard is the rapid eye movement that we make tens of thousands of times a day to change our focus of vision in our environment and scan our environment. This diagram is quite complex, but I've highlighted the frontal and parietal regions where they overlap, and you can see that there are extensive projections all the way down through the centre of the brain that latch onto the neurons that control movement of the eye. It might be a little bit simplistic, but you can actually think of shifting attention as programming an eye movement to look at whatever it is that's captured our attention in the first place. We can then, by looking at eye movement, sensitively measure where our attention is directed by how we control Sicard's. We can manipulate attention using very simple visual stimuli, and I'll show you what that looks like in a moment, and then measuring the response. I wanted to stress here that we're looking at changes to functional networks. I'm not suggesting for a moment, and I'm going to make this crystal clear, that people with visual snow are cognitively impaired. But we are able to measure, using cognitive paradigms, subtle changes across networks, and that is the point of the research. So we start with a very well-defined system, the visual system. We know a great deal about what goes on in the brain in terms of processing vision. There are a lot of gaps, and I'll acknowledge that. We also have a fairly well understood set of processes that modulate activity right the way across that system. And then we have a very sensitive means to interrogate and measure those processes by looking at where our eyes are directed. I'm not going to go into any detail at all here, but simply by presenting simple visual stimulus to manipulate attention, measuring eye movements using a video oculography techniques, or at least that's what we do in our lab, we can create a movement trace that allows us to measure an infinite number of variables, and I've labelled a couple here, but there's many, many more that we can look at to start profiling changes throughout the system. So I'm going to now give you a look at some of the work that we've been doing in individuals with visual snow. The first of these is a paradigm that we use extensively in many, many individuals and across many patient groups also. This is an anti-Sicard paradigm. As I've said here, it's a classic measure of inhibitory control. And I said this was a simple paradigm, and it really, really is. We get participants to sit in front of a screen, get them to focus on a cross at the centre of the screen, and then after a period of delay, we present a cross either on the left or right-hand side of the screen. What we ask our participants to do is not look at that cross and instead make an eye movement in the opposite direction, which sounds particularly simple, but it's actually quite deceptively difficult to perform. So the directive here is to look away from the target. So we're inhibiting a reflexive response, but also facilitating a volitional response. Now, because we've asked the participant not to make an eye movement to a suddenly appearing stimulus, what this does is suppress activity across the whole visual field as a function of shifting attention. So effectively inhibiting attentional capture. As I said before, deceptively difficult to perform. This is just a snapshot of some of the imaging work we've been doing in anti-Sicard. This is not with individuals with visual snow, but what you see is a consistent activation of those frontal and parietal projections to brainstem. And these projections might be stimulus-driven, for example, the cross popping up on screen, as well as cognitively driven. So this is the directive not to make a movement, as well as the directive to make a volitional movement to something that is effectively not there. So I'm hoping you understand this distinction between stimulus-driven versus cognitively driven. So these are the results of some preliminary studies that we've been conducting in individuals with visual snow. So we're looking at 14 individuals. And what we found consistently was that these individuals generated a higher proportion of directional errors. Now, by that I'm saying they moved their eye to the cross, which was not the directive, instead of away from the cross, much more frequently than those individuals without visual snow, which was a very interesting finding. Also, we found significantly shorter Sicard latencies. So they actually made a movement when they did generate a movement much more quickly than people without visual snow. So starting with the idea that this directive was not to look and the allocation of attention should have dampened cortical activity prior to an eye movement, this idea of increased errors means that that cognitively driven stop process, that directive to not make a movement, fails to inhibit quite more often than not a stimulus-driven response. So a response directly to a suddenly appearing cross, a target. With respect to the shorter Sicard latencies, so if this stop process is actually okay, the cognitively driven Sicard is released more quickly, which is quite interesting. Now, what this does imply is hyperexcitability of those neurons that are actually encoding an eye movement, but it also may imply reduced inhibitory control. So we're not solving the puzzle, and we're certainly not able to localise dysfunction with such a simplistic paradigm. This requires a much more sophisticated task. But what it does demonstrate, and I think is quite exciting, is the capacity to measure change across a functional system that in effect has nothing to do with what somebody with visual snow experiences. Just very quickly, I'm going to show you another of the paradigms that we presented. This is slightly more sophisticated, although not very much more so. In this instance, we're asking participants to simply look at the target as it appears on screen. So it pops up in one of these peripheral boxes on screen. What we do prior to presenting the target, though, is present a directional arrow in the middle of the screen. And most of the time, that arrow correctly predicts the location of the upcoming target. Now, what that does is set up a degree of expectation so that when the target does actually pop up where the arrow predicts it's going to pop up, you see shorter latencies than if the target pops up in the opposite direction. So what you see is a transient increase in activity neurons representing the corresponding region, so the region's indicated by the directional arrow. Okay, so once again, we've seen changes that we can measure behaviourally. We didn't see any difference, as I've said here, in directional errors. Quite often with a number of patient populations, what we find are people responding simply in response to the arrow because the setting up of expectation is so compelling that they move before there's even a target. We certainly didn't find that in individuals with visual snow. But what was interesting is we found no effective cue. If you have a look at the graph, the right-hand side of the graph, and I call these control subjects, you see that invalidly cued trials have much longer latencies than those that are validly cued, and that's what you would expect. Based on what I just told you before, this level of expectation, neurons ready to fire versus changing a motor program. You don't see any effect at all in individuals with visual snow, as you can see, and you also see much shorter latencies once again. So we have a really unusual set of behaviours that have seemingly nothing to do with the visual snow phenomenon, but we are tapping into network dysfunction. So what this paradigm does, just to recap, is allocates attention that increases cortical activity in the visual regions corresponding with the arrow. This missing cue effect in visual snow, this is a bit of a mouthful, but they're cognitively driven processes, and we'll call that expectation for want of a better word, is actually insufficient to then modulate a stimulus-driven response. So there's a breakthrough response once that cross pops up on screen. So we're looking at, with a question mark, a hyperexcitable stimulus-driven response. And this also speaks to those shorter latencies to all targets. Again, a hyperexcitable stimulus-driven response. So in summary, from these two very simple paradigms, what we're seeing is the top-down sort of frontoparietal processes are poorly modulating what is effectively a stimulus-driven response. This may also apply to a failure to modulate the perception of what I assume is a virtual visual phenomenon. So what I've given you is a brief snapshot of attentional changes in visual snow. We certainly don't, just from these two paradigms, understand what the mechanisms are driving visual snow, but we're certainly demonstrating consistently a capacity to measure functional change over the networks that are implicated in visual snow. Now, at the moment, our research is fully focused on refining those attentional paradigms. There are any number of ways that we can modulate attention and measure changes across the network. And so what we're doing is devising those paradigms, piloting those paradigms, and we're starting to test using a range of sophisticated paradigms, unlike the ones I've showed you, which are quite simplistic, looking at disentangling this idea of disinhibition from hyperexcitability. And I won't bore you with the ways we're doing that. You just have to trust me that that's possible, as I said, with some sophisticated techniques. We're also dissociating performance in terms of the presence or not of migraine. One of the issues we've had with a lot of the prior research, including our own and the research that I just showed you, that was we have a group of individuals with visual snow, some of whom have corresponding migraine and some don't. So we need to make sure that what we're measuring, all these changes across the network, are not associated with migraine, but are strictly associated with visual snow. But I have to say from my experience, and I have had a look at migraineurs using very similar paradigms, we're seeing a clear dissociation in terms of the behavioural performance. So it's looking quite promising, but hopefully over the next 12 months, and next time we visit for one of these conferences, we'll be able to give you a full attentional profile of visual snow. So the next steps, once we've developed these most appropriate tasks that look at tap into these functional networks, looking at the attentional networks rather, is to pop you guys in a scanner and have a look at what changes, functional changes take place once we provide you with a task that actually taps into the changes that we can measure. And then moving beyond that, have a look at imaging paradigms that look, and I've naffily called them stop and go neurotransmitters, just for brevity's sake. We can also hone in on those regions that have been defined by these attentional paradigms and the complexity that we're hopefully disentangled that can look at the neurotransmitter systems that are guiding these behaviours. And also what's important also is that not only are we helping to provide insight into what's driving visual snow in the first place, but we're providing very sensitive means to monitor change. And the reason that's important is because once we determine what's driving visual snow, we start creating effective treatments. We need to be able to measure the effectiveness of those treatments, and we can do so without simply relying on people like yourselves to say, yes, that's a little bit better, no, that's a little bit worse. We can actually measure these with a great deal of precision. I'm going to leave it there, and I'll hand over to the next speaker, and I look forward very much to receiving some of your questions shortly. Thank you, everyone.